So now let's look at a problem called, we will formulate this problem as two problems. One is a Hamiltonian path problem and another is an Eulerian path problem. Both of them will give you the answer to this problem. We will see, but there are big differences in why we may prefer one over the other. Okay. So how do we represent a genome as a path in a graph? Okay, that is what we are trying to understand here, and then we will be using some graph algorithm to find the path along this genome graph, and then basically get the genome out. Okay, so we construct a graph out of this K-mer compositions, right? So we have this. This is our genome. We are hidden. This is hidden from us, right? What is provided as input is only this, all the three mers here, right? But the genome itself is hidden, but these three mers are all coming from this particular unknown genome that we need to figure out what it is. So you can make them as nodes of a particular graph, right? So you put circles around them, each one of them becomes a node, and then you can add edges based on whether they share a prefix and a suffix, right? Suffix and a prefix, right? So basically, this is how it will be if you knew the ordering of the genome, right? So if you knew the ordering, you know that TAA and AAT, this is the first position here, right? TAA, this is the first position, and AAT is in the second position, ATG is in the third position, and so on, right? All the way till GTT, which is in the final position, right? If you know the ordering, of course, we can construct this. This is a simple path. This is your genome, right? And but of course, we do not know the string, we do not know the ordering, we only have these nodes, that is all, right? So can we construct this genome path without knowing the genome and only from its composition? So that is the question we are asking. And there is one answer to it. Yes, we can, if we simply do these kinds of connections, right? We simply need to connect the K-mer one with the another K-mer if their suffix and prefixes match, if their overlap of K minus 1 matches, right? So for instance, TAA and AAT, the AAA matches between both. So then the suffix and the prefix matches, then you put an arrow from an edge from the suffix to the prefix K-mer, okay? And let's just keep doing that, right? So we have this, right? So this is the original genome, but in addition to that, because we don't have the original genome with us, we also need to do all sorts of other connections, right? So this one, again, please remember that this is hidden, right? And so you have only these three mers that are forming this genome, and so you'll have to make sure that it's not just AAT goes to ATG, but anything that begins with an A, you have to put the link, right? So AAT can go to this ATG or this ATG or this ATG, right? So it can go to this ATG. And is there anything else that starts with an AT? I don't think so. Yeah. So all the three K-mers that start with an AT have been covered here. And then similarly, then from ATG you go to TGT, right? And then no other, and then there is also this TGG, right? So there is TGT, TGG, and then TGC, right? So TGC is already linked up, TGT is linked up, it also needs to go to TGG, right? So because we do not know the original genome, we have to make sure that all pairs of K-mers which have overlapping suffixes and prefixes are paired up this way, okay? That we add edges in the graph this way. And then this is how the overall graph looks like, right? And then of course we do not know the ordering, and I mean the path of the genome is still in there, right? But we do not know the ordering. It's not presented very nicely in this. You can go through the central path and then say that is the genome, right? It is scrambled up, right? So the nodes are really arranged from left to right in this lexicographic order, right? So this is how it is arranged in terms of order in the genome. But then if you scramble it up and then they are arranged in the lexicographic order, then this is how it looks like. All the A's are in the beginning, AAT, ATG, and so on. All those that begin with C's are then, and then finally those that are beginning with T's, okay? So now the question is there a path that starts from one of the nodes and ends in another node and visits all of these nodes? Okay, so that is really the Hamiltonian path problem that you may have encountered before, and that will give you the genome in this graph, right? So by looking at tracing, you need to have a path. A path is really a sequence of vertices following along all the edges in the graph. So you want to start with one node and then you want to keep visiting every node only once until you reach the final node, okay? And you should have visited all of these nodes. for instance, TAA is here, AAT is here. So then you will add, so TAA is here, AAT is the next node, then you will add the letter T as the next letter in the genome, and then you go on to ATG. So then that letter G is added. So this is ATG is the next one. The G is added as the next letter in the genome, and then you keep on going. And so the genome is still there hidden, but it's not presented in a nice way from left to right. Right? You need to be able to figure it out. And let me just run through all of this. So you're picking up one k-mer at a time, and that means you're adding one letter at a time to the genome. Okay. So we are trying to find this path that covers all the nodes in this graph. Okay. So we keep going. So we are walking through the graph. We are finding a path, and we are trying to touch all of these vertices or nodes in the graph. Okay. And this is precisely the definition of a Hamiltonian path in graph theory, a path in a graph that visits each node in the graph exactly once. Okay, that is the definition of a Hamiltonian path. And that will give you the genome like we just saw, because each of these nodes adds one more letter, and then you will get the whole genome sequence. Okay, so the Hamiltonian path is like we just described. It's a series of nodes or a path in a graph that touches every node exactly once. And given an input graph, you want a path visiting every node in the graph exactly once. And so this we want to find out in this k-mer graph that we just built. All right. You can ask questions. I mean, not every graph may have a Hamiltonian path in it, right? So you may ask this question on, does this graph have a Hamiltonian path in it, right? So I'm going to wait for a moment and see if you're able to figure out if this admits a Hamiltonian path. Can you start from a node? So this is one of those stop and think moments that they encourage in this active learning approach. Is a lecture, if it's just given continuously, it becomes harder to absorb. So let's just pose a problem and then think about this problem. Can you start from any node here and then visit every node exactly once? Yes, there are cycles in the graph. Yeah, that is fine. Does that create any issues? You don't have to come back to the original node. You can start with that will become a Hamiltonian cycle problem. That's a traveling salesman is, for instance, you want the salesperson to come back to the original starting city, right? That will be a different version of the problem. Here you start with one node, visit every node exactly once, and end in some other node. Is that possible in this graph? Okay, let me actually show you the answer. It is possible through this series of red arrows, right? You can start with this node, and then you can kind of first trace the inner circle, then you trace the outer circle in a spiral fashion, then you trace the outermost circle, right? And then you can see that this red path, it traces through all the edges in the direction of the edges, and it touches every node exactly once. Okay, it visits every node exactly once. And this is exactly where one of the first people we post in the first slide. I mean, we presented in the first one of the first slides up here. So this was this Icosian game, and so William Hamilton had a uh, role to play in this particular game, and that is the reason also that this is called as an Hamiltonian path problem, right? And so this game has this connection to graph theory and Hamiltonian paths and graphs, right? And you can also think about a problem in an undirected graph, so where every edge you can traverse either of the ways. It doesn't have to be necessarily directed, and that is what this Icosian game here really is. There is, you can see in the board that there are really no arrows in here, right? So these are just the undirected version of this problem. Okay, each edge doesn't have arrows or directions. Okay, so having seen the string reconstruction problem as a Hamiltonian path problem, let's try a different route and then also see if there are other ways to solve this problem. Like I said, it may allow us to solve this problem more efficiently, right? So instead of thinking about 3-mers or k-mers as nodes, let's view them as edges. Okay, that is the way of reformulating the problem. So if you look at this again, right? So here we had 3-mers are nodes, TAA, AAT, and so on from this genome. But can we now put the edges? Can we just add these k-mers as labels to these edges? Okay. And then what do you think we can make the nodes to be? Are there any suggestions? So here the nodes are the 3-mers, and then the edges are just simply connected based on the overlapping suffix and the prefix, right? Here the edges are the 3-mers, and what can we label nodes to be? With which substrings? So the starting letter, you mean the TAA, you will start with T, and then the AAT will be A. Okay, so that is uh, one approach, or actually, oh yeah. So you are saying that you will write down. Okay, let me just write it down. You are saying that you will put T here, you will put an A here, right, and this will be an A, and this will be T, and so on, right? Can you increase the size of this? I mean, instead of the starting letter, can you put more letters so that there is more, and that will make it a little bit easier to formulate the rest of the problem. So that's a good direction, but is there some other way of labeling the nodes? An edge is connecting two nodes, right? And then between two edges you have a node, right? And then in this particular case, let's say we have CCA and CAT, right? So you are suggesting that we put C. Can we add one more letter that makes it more meaningful? CA, right? So we can just we can put something that is overlapping between those two strings, right? So similarly between GGG and GGA, right? So the GG is the overlapping one. I can put tumors, right? So this one I can put GA. Similarly here I can put not T here, but I will rather put TA, okay? And here I will put AA, okay? And this will be AT, because AT is the same between here and here, okay? So this is essentially what we are going to do. We are going to view three mers as edges, and then the nodes we are going to label with two mers, okay? Like K mers as edges and K minus 1 mers as nodes, okay? And the reason is that you want to be able to go from, so this is TA and then this is AA, and then that essentially then will give you the sequence TAA. The prefix of TAA and the suffix of TAA will become the two nodes, and then the K mers will become the edges. So K minus 1 mers will become the nodes. The first step is we are going to take the genome, and then we are trying to convert it into this three mers on edges graph, okay? And then we are going to identify which ones are repeated, and then those repeated ones we used to have it on different nodes, but now we are going to merge them all together, and that turns out to be a key step for us to get efficient algorithms, right? So what we are going to do is that if you see multiple ATs here, right, all of these are these overlapping two mers between at least two K mers in our two three mers in our representation. All of that will come together, right? So this AT, and this AT are essentially the same node. So we should not have three copies of that. We will have just one copy of that. To do that, we will first merge this and this, right? So this is edge E1, let's say, right? So that is this edge E1. And if this edge is E2, right? So then that is this edge E2, right? This edge is E2, right? So you are then merging them. So in the next step, this AT is also merged, okay? All the other edges are kept the same way, okay? And then all of these are merged in this fashion, and you will get this kind of a graph. Again, you can see that there are TGs here. There are three copies of TGs. All of them have to be merged as well, okay? So then all of them are merged over here, and you get this particular graph. And let's see if there are anything else that needs merging. Do you see any other nodes which are copies of each other and we need to merge? GG, yeah. So we need to merge this and this, right? So GG needs to be merged. And you can also allow self edges or these loops, right? So from the same node, you can go back to the same node, because previously this GG had this three merge GGG going to another GG. Once you merge it, then this will have to be labeled on the edge that is looping onto itself, okay? So I think we covered all of the copies. So now this is the final graph, and this is called the Debruijn graph. And again, we will see why it is called the Debruijn graph. Okay, it is named after again a researcher. And so this is the Debruijn graph of this particular genome that we have just mentioned, right? We still haven't specified that if you do not know the genome, what will this graph look like given just the K-mer composition, right? That we need to still figure out. But given a genome, this is how the Debruijn graph of the genome will look like. And again, we ask the question, if somehow someone presents you this Debruijn graph of a particular genome and then asks you, is this genome hiding in this graph, or do I need to know this sequence separately, all right? And again, you can kind of take clue from the Hamiltonian path problem and then say that the same way that Hamiltonian path problem was some kind of path through this graph that visits every node, the genome is again a path through this graph. But what should it be visiting now? Should be visiting every node again, or what should it be visiting? Every edge exactly once, right? So now you allow multiple edges between pairs of nodes. Here you have three ATGs, right? So the k-mer composition is still preserved along all the edges. There were three ATGs in the original genome over here, right? So this ATG, and this ATG, all of that are still preserved in these three regions, right? So we still preserve those k-mer compositions along the edges. So now we want a path that starts from one node and ends in another node and visits every edge exactly once. Okay, so that path is now called a Eulerian path, not a Hamiltonian path anymore. It's called an Eulerian path, and this graph is called a Debruijn graph. Okay, and we will see why these are named this way. Okay, and the genome is hiding in this graph. Yeah, it's a path that covers every edge exactly once, and we can see that now. So we have a TAA, and then you go to the next node from. So this is the first edge you are visiting, right? So first node you are visiting, the second node you are visiting, the third node you are visiting along these edges, right? So E1, E2, and so on, right? So you are going you're going along this E1 edge, that is TAA, then you're going to the E2 edge, that is AAT. You know that when you go from one edge to the other edge through a node, you know that there is an overlapping suffix and a prefix. That always that this AA and this AA will match, right? And so you will go from TAA to AAT. This adds one letter to your genome, and then you keep going. Decide to go this route instead of any other route. Okay, so if we go up, then we take TGC, so that will add a C. Okay, then there is no other way than to take this particular edge, right? So then you will get this CC, another C added, and A added, and so on, right? So now again, here there are two paths you can take. You can either take this path or this path, right? And then we can take any of these paths, and we can continue going, right? And again, you can ask the same question: what are we trying to find in this graph? We already answered that question. We are trying to find a path that will visit each edge exactly once. So we are going down this way, and then finally you go and get this Eulerian path, which is a graph that is a path that visits each edge exactly once. Okay. And every edge that you are adding, you add one more letter, the same way that we did this for the Hamiltonian path. First, let's just summarize what we have seen so far. An Eulerian path problem is you are given a graph and you want to find a path visiting every edge in the graph exactly once. And this graph is a graph that will allow parallel edges between nodes and self loops and so on. So you can have between two nodes, you have these edges. These are called parallel edges, right? So parallel edges. You have three edges here between the node AT and TG. Here you have a self loop. So all of these are allowed in this graph. Given such a graph, we want to find a path visiting every edge in the graph exactly once. And this is exactly the problem that Euler was trying to solve, right? So there was this famous bridges of Königsberg. I'm not sure if I'm pronouncing Königsberg correctly. So this is a small town where you had this kind of landmass in the center, right? You have this landmass, this island in the center, and then there was this waterway that was surrounding the island, and there were these other lands, and there were these seven bridges. So there's 1, 2, 3, 4, 5, 6, 7, right? So you have seven bridges that was connecting different parts of these lands, and there was a problem posed that can you start from a point and then visit all of the bridges, right, without going through a bridge more than once. You have to visit all the bridges exactly once. Okay, you start from a point from some landmass, maybe the inside of the island, and you want to visit all the bridges exactly once and then end somewhere else, right? So this is really the first problem in graph theory, if you will. So this problem was put in as a kind of a island and bridges problem, but Euler was able to see it as an abstract it away and say that you can actually remove all the sizes and shapes and geometry and architecture of these islands. You can just simply think about it as a set of nodes and like points and lines connecting these points, right? It doesn't even matter what the length of the bridges are. And so he abstracted away all the details. He just said that I'm going to label these different land masses with these 4 points, which are the nodes of the graph. And then all of these are parallel edges connecting these different nodes. And then he posed this problem as, does this graph has an Eulerian path? Does this graph have a path that touches or visits every edge exactly once? Okay. So this is the underlying graph, and these are the two bridges connecting the green land and the blue land, and this is the bridge connecting the orange land to the blue land, and so on, right? So and then this bridge. So for each of the bridges, you have 7 edges connecting the corresponding land masses. Okay, this bridge is over here. So then you can abstract away. You just have to look at the graph, okay, and that is where Euler features in. But let's just look at before we look at what the solution Euler proposed for the problem. Let's just compare the two ways in which we have proposed or formulated this problem. One is the Eulerian path problem, and another is the Hamiltonian path problem. In both of these problems, we have the genome hiding as a path in the graph. Okay. And this is like spotting the difference between these problems. If you're computer scientists who know complexity theory, you already know that one problem is much harder to solve than the other problem. Okay, and that is what we will see briefly in the next set of slides. And in the next set of slides, basically, we want to understand how efficiently we can write a code that will solve these problems, right? So how efficient we can design algorithms to solve these problems. And that will also then determine the choice between which problem you want to solve. If you want to write a sequencing assembler that will basically take billions of these reads and then assemble them into a long genome. And the main crux is the following. Euler proposed an algorithm to solve this problem. Even for 7 bridges, he solved it, but it can be scaled up quite easily to even for a city with millions of bridges. And you can say that for a city with millions of bridges, does this city, this configuration of all these bridges admit an Eulerian path or not? If it admits, can you find that Eulerian path? So both of those questions can be answered in very efficient time. And the notion of efficiency in computer science is whether you can devise a polynomial time algorithm, an algorithm that runs in polynomial time in the size of the input, right? You would have seen in your earlier data structures and algorithms courses. So while Euler could solve the Eulerian path problem even for a city with millions of bridges, nobody has developed a fast algorithm for the Hamiltonian path problem yet. It's one of these classic NP hard problems, computationally hard problems.